Before I kick off with the data, there are some considerations to be done, and especially introducing the pathology I will be talking about today. So infantile-onset ascending human spastic paralysis, for now in short I will call it IAHSP. It's an extremely rare disease in the family of the motor neuron diseases. It's a neurological disorder, and it starts from the lower limbs in the childhood, in early age, and then it progressively expands to the upper limbs and to the involuntary musculature. But unlike other pathologies of this family, it's yes affecting the motor neuron axis, but just the first motor neuron. It's an autosomic recessive disease, meaning that it's inherited, it's a genetic disease, and one gets it from two parents which are healthy carrier. The gene which is affected is ALS2, encoding for the protein Alsin, which is a protein which is almost ubiquitously expressed, but just in the central nervous system is, when mutated, it gives rise to the pathology. Alsin itself, it's described to be involved in the vesicular trafficking within the cell, which is a function which is really, really important for the maintenance of dimensionally important cells, such as motor neuron indeed. And yeah, indeed, when the vesicular trafficking generally is impaired, there is a defect in the neuron development and maintenance. Now, what about Alsin? We also have to have a look at the structure and molecular mechanisms. So Alsin is pretty big protein, more than 1,600 residues. There are some domains which are described, especially of note are the RLD domain, which I'm depicted here with the red boxes. It's important for the endosomal localization of the protein. Then there is the DHPH domain, which is extremely important because it's an interaction site for the GTPase Rab5. And that's the key domain to understand Alsin function. There is a so-called VPS9 domain, which is the interaction site for Rab5, another GTPase, which is already since long known to be basically at the crossroads of cellular trafficking mechanism and whose impairment leads to neuron development deficiency. Now here I'm reporting a patient case which harbors two mutations to Alsin, inherited from respectively the parents. The first mutation is not of particular interest, the top one, because it basically gives rise to a truncated form of Alsin which gets degraded. But the second one is a point amino-acid mutation in position 1611 of an arginine, so a positively charged residue with a tryptophan. Important is that in any case, whatever the mutation is, the patient has a version of Alsin which is deficient for the VPS9 domain. And this is extremely important because Alsin in the cytosol is not present as a monomer, but it's present as a tetramer, which starts from the formation of a dimer whereby two VPS9 domains interact, constructing the Alsin dimer, which subsequently tetramerizes by interaction of two dimers. This form is, let's say, the form that is able to recruit active Rab1. And this is important for the localization to lipidic membrane compartments. Then, in a way which is dependent on the functionality of the RLD domain, the tetramer can then go to early endosome and then interact with Rab5. When the VPS9 domain is impaired, all of this is basically not happening, and Alsin is unable to perform its function. So the aims of our work is, first of all, the characterization of key domains which intervene in the pathogenesis of this specific mutation giving rise to IAHSP, the characterization of the structural dynamics, and the repurposing of some already known drugs in order to treat this single patient. Why did I say characterization of key domains? Because the overall structure of Alsin is not experimentally resolved. What's known is the prediction of the structure of the RLD domain, but all the other domains, and especially, and I stress it again, VPS9 is unknown. So to kick off with the data, our starting point was indeed the idea of characterizing the structure of VPS9 domain. Here I'm showing you an extract from a paper from 2004 in cells from Del Prato and colleagues, whereby they resolved the structure of Rab5, which is another VPS9 domain harboring protein, which shows highly homology with a series of other proteins, including, as highlighted by the red arrows here on the left, Alsin. Now, what they showed here is that when one tries to do any alignment of other VPS9 domains with the Rab5, which is the template for a potential homology modeling, there are some gaps. And therefore, the best way to model this is basically to align clusters of sequences, to align just some sequences. And indeed, they break down the structure of the VPS9 domain of Rab5 in three major components: the VPS9 domain core, highlighted in blue here, the so-called helical bundle, and the C-terminal. And after trying to model the overall sequence of the VPS9 domain, we concluded that we had to do the same. So first of all, we did step one of our two-step modeling by modeling the VPS9 core domain for the, of course, wild type and mutated sequence, and then we obtained reasonable models. Later on, we moved to the helical bundle portion, also here obtaining more or less reasonable structures, and then we joined them together by modeling the linker of 10 amino acids, connecting them in the sequence. I have to highlight one analysis that we also performed on these models, and is a CAPS flex analysis, which basically predicts regions which are highly flexible and unstable. And as you can see here from this graph, here I highlighted with an arrow the mutated residue, but overall on the protein structure, there is not such a huge difference, telling us that most likely the mutation is not inducing conformational changes and instability in the structure of the protein, but probably the physicochemical properties of this domain are playing a role in the pathology. Since we know from literature that VPS9 domains dimerize and that Alsin dimerize anyway, we decided to test and predict the dimer structures when trying to dimerize two VPS9 domains, respectively wild type and mutated, and the VPS9 domain, here again, wild type and mutated, with an RLD domain. This consideration is based on some experimental in vitro experiments that show also that the RLD domain is kind of a sticky domain and there's some preponderance to binding other domains, at least in vitro. And as you can see already from these representative structures here, you can see that in the mutated forms, the mutated residue is always at the interface of the interaction. And in this case here, it's also self-interacting. A more thorough analysis, by keeping in mind those mutated forms of all the structures that we got, showed us that indeed the mutated forms overall have always a higher share of tryptophan interacting dimers. Another analysis that we performed is a PDB sum analysis. Here I'm showing you in this panel here with these white dots areas in the space around the protein which are predicted to be of high interest in terms of interaction. And as you can see here, in the mutated VPS9 domain, there is a cluster of interaction areas highlighted by the green oval, exactly in correspondence to the mutated residue, which is in yellow. And this, if one thinks about it, is kind of sensible because we are substituting an arginine with the tryptophan, which might give rise to an hydrophobic surface willing to interact with other proteins. And also another consideration we made starting from this point. If one calculates the solvent accessible surface area here for wild type and mutated VPS9 domain, respectively in the interaction with another VPS9 domain or with an RLD, one sees that always, in this case, the solvent exposed surface area of the interaction with RLD is reduced. So it's pretty sensible, we concluded, that the arginine harboring wild type sequence might be most likely to expose this to the solvent and therefore interact with another VPS9 domain, while we hypothesized that the mutated one would prefer to interact with the RLD domain. And indeed, when we characterize our system with the amber force field and calculated the interaction energy, we obtained this nice trend showing us that indeed the VPS9 mutated domain interacting with the RLD has the highest interaction potential. Another analysis that confirmed this hypothesis was to utilize the Calderaro model published in 2020 for the calculation of delta-delta G, measuring instability of a protein complex when a mutation occurs to one residue. And indeed, also by doing this, we obtain that the VPS9 domain, the mutated form interacting with RLD, looks more stable, highly stable, when compared to all the other forms. So with this in mind, we are currently organizing and planning to perform a steered molecular dynamics simulation in order to furtherly test our condition and representative structures. So the idea is to apply a force to both systems, analyzing the trajectories and seeing whether the VPS9 mutated domain interacting with RLD looks more stable and resistant to the changing. But I also have to mention, we are optimizing that with NAM2. We are currently encountering some issues in doing that. So if later on or in the social event, anyone has experience with this type of simulation and their suggestions, they will be extremely welcome and thankful. But moving on, we also had in mind to have a pharmacological strategy in order to treat our patient. So by having the model, the mutated model, we defined a region of within 5 angstrom of some residues of interest that might be the target, basically, of a ligand search. We individuated the pharmacophore with pocket query. We utilized 3 different online tools to individuate a cavity, and all the 3 individuated a similar cavity in proximity to the mutated residue. And therefore, we move further on, and we perform a structure-based virtual screening by having as a query the cavity that we had found. We screened about 10,000 drug-like compounds. We filtered the highest hits for blood-brain barrier permeability models. And the results is shown here on the right. So in purple here and the green arrow, you have the mutated residue. And as you can see, the best hits that we have found are somehow sitting around in the cavity like some sort of scarf that in our idea is modifying the physicochemical properties of this surface. The idea is then that by applying these molecules, we can change the interaction of the VPS9 domain, which prefers, we have been demonstrating that, for the RLD, re-establishing the correct dimerization. I also have to mention that the best candidate that we are having is currently under promising preclinical experimentation, and of course we cross our finger for the definitive data. Another aspect that we had to take into account is the interference of our drug strategy and anyway the mutated residue with the Rab5 activity of the protein. And to do that, we had to model the interaction of the VPS9 domain with Rab5. In the PDB, there is a structure in arabinosis thaliana of Rab5 in complex with the VPS9 of RabX5. There is high homology with the human proteins. So by superposing, we obtained a series of interaction models. Here I'm depicting a representative example that shows us by simple visual inspection that the mutated residue, here highlighted in green, is totally on the other surface of the protein, then on the other face, than the interaction surface with Rab5. So supposedly there is no interference. Moreover, a literature search showed that some residues have been shown to be essential for the Rab5 activity. Okay, and indeed, 4 of them are conserved in the human VPS9 of Alsin, and none of them is in close contact, neither with the mutated residue, nor with the residues interacting with our drug candidate. So here I'm summarizing more or less our findings. We know, it's known that alsin in the cytosol is able to dimerize through interaction of the VPS9 domain. And then subsequently it tetramerizes. And this allows it to recruit active RAC1, localize to membrane ruffles, subsequently be included in early endosome thanks to a functional RLD domain. And we also demonstrated here that the mutated alsin with the mutated VPS9 is abnormally dimerizing, impeding the further development of this process. What we have done with the screening is to individuate the drug candidate binding the mutated portion of the VPS9, displacing the aberrant interaction, the aberrant dimerization, and supposedly re-establishing the physiologic function. With that, I'm already coming to the acknowledgements. I would like to thank my group, for sure, Professor Caron and Hermon is supporting me and giving me the chance to work for that. Sara Solaro, a brilliant student of us, The Porteous Lab, so Dr. Soares and Professor Porteous for sharing the RLD homology model that they have been publishing and nicely shared with us. All of you for the opportunity and for listening to me, and especially help Polly, the foundation, which is supporting this research. I will be open to question.